So welcome everyone to this 0PM webinar. Today I am having the pleasure of introducing the webinar, and for those of you who don't know me, I'm Sarah Hale. I work for the German Water Centre, and together with Hans-Peter, who's here as well, we're leading 0PM. Today we are having a webinar from two of my really good colleagues, Emma Knight and Ian Allan. Emma works for NGI at the same place as Hans-Peter. She's got lots of expertise with chemical pollution monitoring and the use of passive sampling. And Ian Allan also works in Norway at NIVA. He's had vast experience with passive sampling, I would say, using it as a tool for risk assessment and to be put into regulation. Today's webinar is all about passive sampling. So what we're going to be hearing about today is how we can use passive sampling as a tool to support monitoring efforts. We're also going to look at what we've done in 0PM because we've used passive samplers in different locations. And then we will see how we could possibly fit passive samplers and results into policy and regulatory frameworks. The webinar is part of the 0PM project, so I assume everybody knows about the project, but what we do in 0PM is we interlink prevention, prioritization and removal strategies to protect human health and the environment from persistent mobile toxic and very persistent very mobile substances. What I will say is during the webinar, if you have any questions, please pop them in the Q&A box, and then at the end, once Emma and Ian have spoken, Hans-Peter will run through the questions. If we run out of time and you've left your name, if it's possible, then we'll follow up with you via email afterwards. We'll share the slides and we'll share the recording so that you can listen again. And I think, without further ado, I can pass it over to Emma and she can share her slide and get started.
Yeah, thank you very much. I'll just share my screen. So hi everybody. Thank you very much for the introduction, Sarah and HP. So we're going to try and look at beyond grab samples, passive sampling for smarter environmental monitoring. And as Sarah has maybe already said, we're going to look at what is passive sampling, how is it used, what can it measure, practice within the 0PM project, and how we can integrate passive sampling better into regulation. So what is passive sampling and how is it measured? So passive sampling is based on a time-integrated diffusion-based accumulation of the freely dissolved contaminants into a device or a sorbent or receiving phase, allowing for an estimation of average concentrations over the deployment or exposure time. And this can be in both in situ and like ex situ in the lab to determine the C-free concentration. So that includes in air, water, and sediments as well. So why passive sampling? Well, we have this continuous monitoring period. So it could be days to weeks that will give us this average time-weighted concentration. Applied or applicable for many different matrices. Again, can measure this freely dissolved concentration in the contaminant over time or in the matrix over time. Can be related to bioaccumulation in organisms because we're looking at this C-free concentration, and we will talk about this more later on as well. There's also both linear or first uptake rate kinetics that we'll look into. So it can also be at equilibrium concentrations measured in a polymer, can be proportional to the chemical activity in the medium being sampled. And activities in different media can therefore be more can be compared on a polymer basis. So when we look at the principle of passive sampling, it's absorption and diffusion process controlled by the uptake. So we have a concentration that's in the water of our chemical of interest. We have this water boundary layer that there's slowly a diffusion across this water boundary layer into the membrane of our passive sampler or across the membrane of our passive sampler. Then we have this same pattern when we look at the membrane to the sorbent. So the sorbent is the receiving phase of the passive sampler that will collect the contaminant of interest that we're looking for. And when we look at this in terms of first order uptake kinetics, we have two phases that we are usually looking at, and that's one in this equilibrium phase, so n equal, or in this linear uptake phase. So over time, there'll be a mass accumulated in the sampler, and this can usually be in a linear uptake phase or in an equilibrium phase, although there are some that we measure in between. So up over to Ian from this point.
Yeah, hi everyone. I'll take over from this point. So the next couple of slides are to present essentially the practical steps for passive sampling. So you have an idea of the chemicals you're interested in, and then the first step is to select the sampler that will be best suited to the application that you have. And then it's a sampling trip to a river, to wherever you're planning to deploy your sampler. You see, actually, the Smurf here is very smiley going out in the field, and Emma was very smiley as well. And then you leave the samplers for 1 day, for 1 month in the field. You come back to your deployment site and then collect the samplers, usually with some form of fouling or something on them, like that. And then you bring back the samplers to the lab, and then the next step is you dismantle the samplers. So Emma actually made these very good videos for showing how to do it. We try to focus when developing a sampler on something that can be relatively straightforward to implement in the lab and in the field. And there's no point doing things too complicated if we cannot ultimately bring it into a sort of routine use. And here we see the dismantling of a POCUS sampler. So this one has got a stainless steel mesh membrane. We see that here we take the two washers apart, and then we're left with the two stainless steel mesh, and then we separate these two, and we find the sorbent disc underneath, and that's what we will collect to place in a beaker or a jar for extraction. Yeah, we and then there's an extraction and analysis, and then some data interpretation to be able to go from knowing the concentration of the chemical you're interested in the extract that you've analyzed, back to a concentration in water for the exposure site that you monitored. Yeah, so the advantages of passive sampling. So it's a methodology that is robust, has got a good repeatability and reproducibility. We know that when we deploy silicon samplers simultaneously, we have on the masses accumulated, we are down to around 10% or below in terms of variability between replicate samplers. So we know that we've got something that is reproducible. And it also minimizes sampling artifacts and sampling and handling artifacts. So because we have prepared a sampler that is made exactly in the same way as the exposed sampler, we've got actually a very good blank. So these are the key advantages of the operation of the samplers. So high reproducibility, that's actually very good for trend monitoring, and it's also very good for if you have some sites that you would like to compare in terms of concentration. It integrates changes in concentration over time, and as Emma mentioned to start with, and this is basically incorporated into the average concentration that is calculated at the end. When applied to sediments, the samplers can help you to measure freely dissolved concentration in sediments. It can also help with looking at the accessibility of of chemicals in these matrices. And that is quite useful in situations of contaminated sediment risk assessment and remediation. The C3 is directly linked to organism exposure, toxicity, and bioaccumulation. So in terms of environmental risk assessment, there is definitely a place for passive samplers. And another advantage is essentially that they don't need electricity, active pumping, and so on. So if you're planning for some passive sampler or some measurements at a remote sampling site, like working in polar regions, deep water sites, this is actually a useful methodology as well. And then the second step, we'll look at passive samplers and what they measure, and we'll look at an example concrete application before we move to looking at some of the things that have been done as part of ZeroPM. So in this table, we can see the samplers and the measurements are organized through the matrix that is sampled. So air sampling has been conducted quite a bit with polyurethane foam discs and sorbent impregnated puff discs, and that's been to measure gases, but also particulate bound chemicals for the particulate that settles in the puff matrix. The deployments are mostly in situ, and that's the same for water, measuring freely dissolved chemicals in water. So when we're looking at non-polar, non-ionized chemicals, we're using low-density polyethylene, SPMDs, which are LDP with triolein lipids inside, and then silicon membrane. And then the DGT chem catcher and POCUS devices have been developed and used mainly for relatively more polar chemicals and ionized ones as well. The DGT, which stands for diffusion gradient in thin film device, was originally developed for sampling labile metals in water and sediments. And then we have exposures in sediment and soils. That's interesting because it can be done in situ in sediments, but it can also be done ex situ in the lab, bringing your sediment back, and you can measure a C3. It's also possible measuring a C3 to be able to, with total concentration, to look at octanol water and not organic carbon water partition coefficients, for example. But the tests can also be optimized to be able to look at that accessible fraction of and easily dissolvable chemicals in these solid matrices. LDP and silicon are mainly used for looking at these non-polar chemicals, but there's also been groups of people working with 10X and XAD sorbents or DGT in some cases. And with the DGT, you can also, with multiple deployments alongside, you can also look at desorption rates. And then the final matrix is biota. That's been done a little bit mainly ex situ. It's activity-based equilibrium sampling in tissues. So there's been applications to marine mammal blubber, to fish muscle, liver, and internal cavity of fish. And basically it involves bringing a concentration of the chemical of interest at equilibrium in the polymer with the tissue of the organism that was sampled. On this slide, you can see the sort of typical deployment times for the different samplers. So the MPT that has been used a lot for PFAS, DGT, ChemCatcher, and POCUS, the deployment times are usually no more than about a month. In the case of DGT, for example, when using agarose gel, the gel will tend to not last much more than this kind of time. Sampler like SPMD, silicon, LDP can be left a lot longer to sample. And we've had deployments up to about 2 years at deep water sites in the marine environment. And then the next few slides are going to be an example of passive sampling that we've worked with. That's to try to apply it in support of biota monitoring for the EU Water Framework Directive. So that's work that and a lot of this effort to join some data sets that have been acquired through different projects was helped through the Norman network. So we have passive sampling for hydrophobic and non-ionized chemicals through absorption-based devices, and that's a pretty well developed methodology. The performance reference compound-based estimation of sampling rates, RS in situ, is pretty robust now and has been used by quite a few labs across the world. There's a good availability of polymer water partition coefficients, these KPWs, for a wide variety of chemicals. And there's also quite a good list of lipid polymer partition coefficients for these same chemicals. And that's very useful because that can help to interpret some of the data in the way that I'll present in a couple of minutes. And finally, over the last decade, there's been a number of quasi-MEM intercomparison studies that have been performed. So the data to support the implementation is really there. So yeah, so in an ideal, or not ideal, but in a situation where all the phases, whether it would be passive sampling or whether it's the fish, if all the phases were at equilibrium, the actual activity in all the different compartments would actually be the same. So this is a hypothetical situation because the fish is not likely to be at equilibrium. In the case of a fish that's sitting at a quite higher trophic level, then in case of biomagnification, that level will tend to go up, and depending on how hydrophobic and how fast the chemicals accumulate in the passive sampler, they could actually be at equilibrium with the water, or they could still be very far from equilibrium. But that's the starting basis for the comparison that we've tried to make here. Next slide, please. So what we've tried to do is to assemble data sets that existed on fish passive sampler comparison. So the first step here in this comparison, we've got absorption-based passive sampling being done with silicon rubber performance reference compound to estimate C3. In all cases, the same partition coefficients were used for the same for each of the chemicals that we looked at. Then we can calculate, we've got a C3, we calculate a concentration in silicon at equilibrium with the water. And then from that concentration in silicon, we calculate a concentration in lipids that essentially would be at equilibrium with the water that we've sampled with our sampler. So from the passive sampling in water, we end up with a concentration in lipids that would be at equilibrium with that water. And then we take the fish data, concentrations in the fish on the lipid basis, and then we can try to compare these two values to see simply how they compare. And then we'll see that on the next slides. So we tried to assemble data that existed through different studies conducted at different sites. And here we've got hexachlorobenzene, HCB on the left, and then we've got BDE47, polybrominated diphenyl ether on the right. The scale, the y-axis, is essentially the activity in the fish divided by the activity in water. So what it means is a number that's above 1 is telling you that the fish is more concentrated than the water. And then a number that's below 1 is telling you that the fish is not as contaminated as the water is. And that's very interesting because hexachlorobenzene, all the data is sitting not so far from the one-to-one relationship. There's a few numbers that are above 1, but most of the data points are below or at around 1. I forgot to mention the x-axis is the trophic level of the fish that was sampled, either measured or assumed. And then for BD47, the picture is even more striking because actually the concentrations in fish could be as low as 2 orders of magnitude below the concentration level or the activity level in the water. So what we tried to say with this is okay, it seems that in most cases the fish is never more contaminated than the water. So in these conditions, the passive sampler is actually giving us a conservative estimate of what would be in the fish swimming in the water that we've sampled with the passive samplers. So let's go to the next slide. So what we propose with on this one, so we're taking these 2 compounds. We've got specifically for the Oslo Fjord, we've got the cod data. These are the box plots, and then the blue lines are the concentration in the calculated from the silicon, and then we've got the EQS biota. So it's essentially taking the graph that we had on the previous slide and then added the EQS biota onto that. And then we see that for HCB, if we use the passive sampler instead of a fish, we would have said that, okay, we're far off the EQS. In the second case for BD47, we see that the EQS is actually a lot lower. The passive sampler are still telling us a conservative value of what's in the fish, but it's also, if we had a tiered approach using a passive sampler first, here the passive sampler would tell us, well, okay, here with these levels, you're likely to be above the EQS, so then you have to implement biota sampling. So this is a proposal for using samplers together with biota sampling. And then, and that's sort of the procedure we're proposing. Annika Janke made this very nice picture. So passive sampling in water, we estimate the concentration in lipids. We have a trigger value, whether that's a EQS or a value according to the EQS. If we pass that trigger value, then we have to implement dedicated biota sampling and analysis. But if we're below or well below that trigger value, then we don't have to do biota sampling. So that's at least one way we think that passive samplers for such substances could be used.
Okay, great, Ian. I'll take over from this section. So passive samplers in practice within the ZeroPM project. So for ZeroPM, we have developed passive samplers as part of the project to test for persistent and mobile substances. And we've also done this at the 3 test sites in the ZeroPM project. So 2 of these test sites are in Germany, they're drinking water treatment plants that take groundwater that has been contaminated by various different chemicals and then treats it before sending it out to the public. And in Greece, it's a wastewater treatment plant that is, it's a wastewater treatment plant. So I'll talk a little bit about what we've done as part of ZeroPM with passive sampling at least one of these sites, and just show you how the results compare to typical water sampling and how we can use these measurements in the future. So for the passive sampling development within ZeroPM, we've used a polar organic chemical integrative sampler, and we've done a number of different steps to be able to optimize the sampler before we get to a monitoring stage. So this includes membrane transfer. So when we have that outer membrane before the receiving phase, how does that react with the chemicals that we're looking at, and does it, what is the water boundary layer effects of that membrane? We need to know this to be able to gauge what our sampling rate of the sampler will be. The same with the affinity and capacity. So this is the receiving phase or the sorbent phase, and we usually do a batch equilibrium experiment to get a KD value or a KS value. And then we can calibrate this in the lab. So we can do a time series of samplers that can be taken out at days 1, 2, 7, 14, and 28, let's say. And from there, we can get an idea of the uptake rate or our sampling rates, RS. And from there, we can do some monitoring. We do find that one type of sampler doesn't fit all scenarios. So it depends on the type of matrix that you want to sample and the type of chemicals you want to sample, for instance. But we have done some field deployments in 2024 at the German Waterworks, and that's what we'll get into now. And supplementary to that is monitoring using passive sampling can also be a way of source tracking the chemicals that we're finding in the environment. So if we go upstream, is there a presence or an absence of these chemicals? And we'll also talk about this once we get to the policy integration section. So we did field deployments in Germany. These were 3 weeks. They were in 2 different drinking water treatment plants, so Rastatt and the Upper Rhine. These are the 2 test sites in ZeroPM project. We deployed 3 different types of passive samplers, so they may have different uptake rates. They may sample slightly differently. The water sampling at deployment and retrieval, so this was grab water sampling to get an average concentration of the water from a grab sample versus what we might see in the passive sampler. And then also to help us understand the flow conditions in the deployment, we spiked some silicon with performance reference compounds to aid in the understanding of our sampler. And in terms of the Rastatt deployment, we did 2 samplers in each of the red starred locations. So this was after the groundwater inlet, after the activated carbon filters, and then also after the pilot treatment plant, the pilot treatments. So in this case, I'd also like to mention that there's 3 activated carbon filters here, but in rotation at the facility, there is 4, with 1 always being out of rotation, and they all have different ages that the activated carbon has been in residence for. And this will be important in a minute. So when we look at a comparison of our grab sampling versus our passive sampling. So firstly, this is the grab water sampling. So this is the concentration measured in the water from the grab samples in the small wells, the inlet, the 3 activating carbon filters, and then the pilot treatment. For this is for the perfluorocarboxylic acids in particular, but we have measured other contaminants at the other drinking water treatment plant. And then this is the mass accumulated in the passive sampler over the 3-week deployment time, also for the perfluorocarboxylic acids. And what I really want to take here is that the mass of PFAS accumulated in the passive sampler is relative to the concentration measured in the grab sample. And we see this in like the profile of the PFAS that we see. So if we put this red bar across the small wells, this same pattern when we put it over the small wells for the posties, even though they're not directly comparable in terms of concentrations, we see this same pattern. We see the same relative profile where we see a lot of PFOA and a little bit less of the PFBA. Secondly, we can also see more in the passive sampler extracts than we do in the grab water sampling. And this is because we get a lower limit of detection, detection of our analytes in the passive sampler, because they've been there for longer, they absorbed more of this average concentration over time. And so when we look at this, and yes, the bars are very small, but we see in activated carbon filter 4, we see a little bit of PFBA. In the pilot treatment, the pilot treatment steps for the activated carbon plus the ion exchange filter, we see a little bit of HPA, where we don't see that in the concentration from the grab sample. And as expected, with an increase in treatment step, we see an increase in or a decrease in our concentration measured. So that's great, because we don't want to be sending out contaminated water to the public. So what does that look like when we back calculate the concentration in the passive sampler to an actual concentration in water? And how is that relative to the measured concentration from the grab sample? So as we can see here, this is when we've made the concentration in the passive sampler relative to the concentration in the water and not just mass accumulated. And what we're looking at is the bars, which are the passive sampler, versus the dots, which are the concentration from the grab sample in water. So the dots and the bars, they match up very, very nicely. And this is fantastic, because then we can show that our passive sampler is working how we hope that it should be working. And very briefly, we can use this calculation from the calibration to then calculate, that's how we've calculated our concentration in the water from the mass accumulated. So then policy integration. So what does passive sampling look like when we look at policy? How does it work? How does it fit in? So firstly, how does the EU Water Framework Directive frame passive sampling? Well, firstly, there's nothing prohibiting the use of passive sampling for regulatory monitoring. So that's fantastic. And I think second to that is there's two important statements in the Water Framework Directive. One is that there should be systematic and comparable results or basis throughout the community, throughout member states, which I think passive sampling can really be useful for. And then secondly, the EU Parliament and the Council should, on proposal for action for the Commission, agree on substances to be considered for action as a priority. This means that the priority list is dynamic and can change over time, and therefore how we measure and respond might also change over time. And so we'll get into this a little bit. So in terms of comparability, and we've seen this a little bit with what Ian has presented, firstly, if we look at biota in a fish species from Spain versus a fish species in Norway, different countries, they're going to be completely different fish. They're going to have different concentrations, different environmental conditions. They could also be at different trophic levels. So then we have another difference that is hard to relate in terms of comparability. And we see this in terms of there's trophic correction factors, but these have a high uncertainty. And this is where passive sampling is independent of the food web. It's independent of of some of the factors that are contributing to unreliable and comparable results. So then we all can see the same thing in sediment and soil monitoring, where they're usually compared based on a normalization of the organic carbon content, which doesn't reflect bioavailability. So you might have a soil or a sediment that has a very high organic carbon content, but the exposure in that soil is actually very low. Therefore, comparability doesn't reflect bioavailability. And this is also where passive sampling can be very beneficial, because it measures this sea-free portion directly. So passive sampling mitigates issues with differences in species, trophic variability, and uncertain correction factors, giving us a more reliable comparability, and that's more in line with the Water Framework Directive. And this is because passive sampling operates in effectively what I would call bioavailability mode. So when we look at this figure here from Ortega Calvo in 2015, where this was proposed as a way to help scientific compartments for a chemical in in a matrix to be put into regulation or to be adopted by regulation, where the chemical activity is measured by passive sampling, for instance, which is in this dissolved concentration phase, so in the orange box, but also could be a little bit in the purple box. And it's directly related or directly in contact with organisms or plants or in this dissolved fraction. And this chemical activity is independent of sediment composition, organic carbon content, grain size, and species differences, meaning that passive sampling can be more comparable. And then if we were to look at this in terms of a tiered risk assessment or management framework, often we are looking at, or what's considered, is this total concentrations versus what we might actually expect to be bioavailable. So exposure might be different based on if you measured in total concentrations or if you measured in bioavailability. So when we look at a tiered risk assessment, often we have this generic risk assessment that looks at total extractable concentrations, whereas passive sampling probably fits more in this tier 2, where we look at bioavailable concentrations. And bioavailability links to fusion, bioaccumulation, toxicity, and risk altogether in terms of that risk assessment. And when we look at the potential to add new chemicals to a priority list, this might then be from passive sampling that could be used as an early warning system when integrated, especially with suspect or non-target screening. So we might see a chemical that suddenly is appearing where it was not before, source tracking, which we talked about before, but also forensics, so wastewater-based epidemiology, for instance, or in potentially in criminal activities. All right, thank you.
Yep, I'll take over for the last few slides. Yeah, and to finish on integration of passive samplers in legislation. So passive sampling has been mentioned as a complementary tool in a number of the EU Common Implementation Strategy guidance documents. It's been mentioned, but that actually dates from quite a while in the Norwegian sediment risk assessment guidelines to be able to measure KOC values, to look at leaching from sediments and looking at the risk for the chemicals moving from that contaminated sediment or accumulating in organisms living in that sediment. And then that's an example from actually 2000, the Australian ANZAC water quality guidelines, for example, for metals, were recommending to look at the dissolved fraction because that was what was considered bioavailable. So they were recommending to compare the concentration of dissolved metals to these DGV values using either speciation modeling, so for example, using the biotic ligand model, or using passive samplers such as the DGT, which will measure the labile fraction of metals in the environment. So that was a good example of trying to promote the use of these tools in these settings. And then the final one is that actually the legislation in France opened up for the possibility for using passive samplers for surveillance monitoring as part of the WFT. So that was also good progress. So there has been quite a bit of discussion over the years for how to bridge, how to get to that next step of using samplers in a much more regulatory setting. There may be a need for standardized use, or at least much clearer guidelines on which tool to use in which conditions. We need to be sure that people understand what is being measured, whether it's bioavailable, whether it's a C3 or a total concentration, have to remember that in some cases EQS is environmental quality standards are derived from ecotox studies, and these are essentially developed with looking at toxicity or accumulation, but with the chemicals quite often dissolved. So are we looking at total? Are we looking at dissolved here? And therefore we need to align the data from the passive samplers with environmental quality standards. We need to be able to get more data from inter-laboratory validation and accreditation. So in that case, there's a couple of very good examples. So there's a number of papers that have been published with Case Boyd that's been really pushing this through Quasimem, looking at the inter-laboratory performance of silicons, and then lately for the more polar compounds. And then Shiel Jonker published in 2020 a Nature Protocol study. I think, Sarah, you are a co-author of that paper that is showing how and showing a clear procedure to measure C3 in sediment. And these are the kind of steps that we really need to for implementation. And then we need a QA/QC that's consistent with the regulatory compliance. So that's having the correct limits of quantification and measurement uncertainty. Measurement uncertainty, that's interesting because if you're using a passive sampler for sampling over time, so you have to also compare with the measurement uncertainty of your bottle sampling from one or multiple sampling events. And then some policy needs developing some of these technical guidance document, and that could be done through the working group chemicals. So trying to develop a guidance document for using passive samplers rather than having them mentioned as complementary tools. We need also reference methods, perhaps adopted by standardization bodies. So, but we do have the ISO standard for passive sampling that can be at least used to say we follow these guidelines. So ultimately, regulators need defensible and harmonized methodologies to be able to have the use of these tools in regulatory monitoring. Yeah, governance and institutional integration. There's a number of key actors here for using and applying passive samplers. And then what we feel there is a need for is to continue with demonstration projects. So this we've had some examples through the Norman network. There was a study in France with AquaRef to look at the quite wide demonstration of using passive samplers in a wide range of water bodies throughout France. And then so that we need ultimately we need this, and then we also need to be able to develop the data set. So we presented some passive sampling alongside fish, but actually we need to increase the size of these data sets to be able to convince people that there is a way to use these tools properly. And then try to embed, when we can, passive sampling in monitoring programs in RBMPs at the next round, and then try to apply it in situations like, for example, when looking at watch list chemicals. And then, yeah, ultimately, we need further regulatory acceptance. We need to develop, this is really a conclusion slide, we need to develop technical guidances, have demonstration projects. We really need to take advantage of the potential of passive sampling, that's having data comparability, especially across wide scale, and to have a cross-agency or national EU-wide harmonization of their use. And I hope the message is I think quite clear, that I think the science is really ready. Now we need to be able to find the right way to put them into into use. Thank you.
Great. And just the last slide to thank all of our ZeroPM partners working with us as part of the project. Yeah. And some references. Thank you.
That was great. Thank you very much, Emma and Ian. Perfect. Now we're going to take the questions from the Q&A box. So if anyone has any, I see there's a few in there, but if anyone has any more, please pop them in, and Hans-Peter's going to run through them. Okay.
Great. That was great. Thanks, Sarah. And hi, once again, I'm Hans-Peter from the Norwegian Geotechnical Institute. And thanks, Emma and Ian, for that tour de force on how passive sampling can be used for smarter environmental monitoring. As Emma, you said, on bioavailability mode, and that's where we want to be. Yeah, so you cover a lot of ground, some key principles, field trials. I was particularly interested in your policy and regulatory framework recommendations, considering it's a bioavailable metric. I'm curious to see if anybody wants to bring that up in the Q&A. And of course, your way forward that we should take more advantages of passive sampling, and we need things like accreditation and standardized methods to make this happen. So with that, I will look into the Q&A box and see things as I am, what has popped up. And I think it'll be kind of in order. So the first questions will be from the first part of the presentation, and the Q&A box will be open until we close this in about 13 minutes. So we'll see how far we can get. All right, so the first, the first question is an interesting one. It would be a different principle, I think, but can you measure microplastics and nanoplastics with passive samplers? Anyone want to answer that?
Great question. Thank you. I think definitely we could measure plastic chemicals using passive samplers. That is definitely one route that we can look at how plastics in the environment. In terms of microplastics and nanoplastics physically, I mean, I don't think it's impossible, but well, for passive samplers, probably. It's not probably ideal. Maybe there's some formal way to make it like a little capturing device or something, but you're still going to get other like particles and stuff in it. So I think for chemicals, yes. For physical plastics, perhaps not.
Yeah. No, and I think also conceptually that's like a different type of sampler. That's like a deposition sampler, particle sampler. So it wouldn't rely on chemical diffusion. Yeah, no, but thanks. Okay, just from next question, how fast does diffusion into passive samplers occur? It of course deviates because of the media, but for example, water sediment, maybe you just need some approximate time ranges for water sediment, maybe for in-situ deployment.
Maybe Ian, you'd like to answer?
Yeah, it really depends on the conditions. So the sampling rates, if we are in a river or marine environment, will be in the order of with silicon samplers will be between 5 and let's say 20 liters per day extracted equivalent. If you're using a POCUS, it'll be 100 mLs, give or take, and that's the sort of range. But if you work in the lab, particularly with sediments, you can actually equilibrate a polymer with sediments by optimizing the kinetics of uptake by shaking more or less thoroughly the sediment, and so for a let's say PCB 180 will take years to reach equilibrium with the silicon deployed in the field. You will be able to reach an equilibrium in the lab under with an exposure in sediments within maybe a month or so. What you need to be able to do here is to make sure that you don't have a depletion of the sediment. So what you're optimizing is the amount of polymer that you're using versus the amount of sediment to be able to have a non-depletive system.
Great. Thanks. And next question, it's missing a bit of context, but I think it was from one of your slides, Ian. It was from Peter van der Oor. It starts, this is not equal to a BCF, seems to be quite low. I might need some context what slide that was referring to. Do you recognize it? Or Peter, you can put in the Q&A maybe a clarification. If not, I'll just go to the next question. We can come back. From Christina Mraz. Have you observed significant differences in uptake when passive samplers are deployed at different water depths at the same location? So like a water depth profile. I'm assuming maybe different systems, river or lake or anything like this.
Not, we haven't had, or have not had that that kind of experience. What we've seen a couple of times is, especially in Norway, when there's sunlight and very clear water. In some cases, if the samplers are not shaded from the sun, we've actually seen photodegradation of the chemicals and the deuterated pHs that we use as PRCs for the samplers that were deployed the closest to the water surface. But we didn't see that deeper. The deepest deployment we've done was north of Svalbard for 2 years. And the samplers came out pretty much as clean as they left. So that was very interesting. Very, very little biofouling on the samplers. And I guess there's at least there would be less potential for differences in sampling rates because of that.
Great. And next will come first a comment and a question from Alexander Scott. The comment was on the microplastic discussion. Micro nanoplastics, you should be able to measure. The nuclear industry has used tacky shades to collect airborne particles, which sounds like a similar principle. Okay, thanks for that. And the question is, can passive samplers distinguish rate effects or only the total over time? Could it tell the difference between a location with consistently high level and a location with a normally low level, but a single event spike? Yeah. I guess that's about time integration.
Yeah. If I understand the question correctly, it's a bit difficult probably for a passive sampler to pick up an individual spike in contamination unless it happened to be over the exact period that you had of deployment. It can definitely tell you the average concentration over time, and I think that is quite important compared to taking a grab sample at one specific time, because you have such fluctuations in variability from month to month to season to even during the day or during the week of different types of chemicals. I mean, particularly if we looked at like pharmaceuticals in the water, for instance. But usually a single event spike will be likely difficult for a passive sampler to pick up, from what I understand.
Yeah. No, and I would just come in, I would just say, and I think if you had a very well calibrated model in a system and you saw a concentration in your passive sampler, you could either say, this is from a constant concentration over this time window, or you can model different types of fluctuations that theoretically would have given you the same concentration, including a single event spike. Yeah. But you would, yeah, but it would be a form of time integration within whatever the diffusion kinetics in the sample are. Okay, an anonymous attendee asks, what do you recommend for future practice and use of grab sampling, passive sampling, and biota? The key point seems to be one of understanding and managing uncertainty and determining purpose of monitoring, ecological relevance of measurement, cost effectiveness, and statistical power. There are many chemicals in our waters, far more than covered by the Water Framework Directive priority substances. So how do you capture a wide range of unknown unknowns? I'm gonna maybe you can deflect that with non-target analysis, but feel free to answer that.
Yeah, I think combining at least to address the unknowns, combining passive sampling with non-target is a good starting point. Mm-hmm. It's particularly interesting if you're looking at, for example, the source at two environments that are under pressure from different, so relatively pristine, or if you're looking at upstream and downstream of the wastewater treatment plants, then you have a good basis of samples to look at, okay, what are the features that have changed from sampling that environment to that environment, and then you can dig into these data a bit more. In some cases also because you're sampling continuously over time and maybe with better limits of or accumulating larger amount of chemicals, it might actually help with that step of characterization and elucidation of the chemicals that are there.
Mm-hmm. Yeah, no, and I would agree with that. And I also say the passive sampling, since you're sampling in a polymer, it's also you also have kind of a pre-extraction step for the chemical space domain that's good for that sampler too. So it also helps if you're doing, for instance, non-target analysis as an initial pre-extraction.
True, there's a form of selectivity. It's already, so yeah.
Great. Okay, from Xavier Weiten. One more good reason to promote passive samplers. The growing number of analytical parameters to monitor is very biota consuming. So electric fishing cannot be used limitless in little river basins. Correct. It's environmentally friendly. Annoying. Thanks for the nice presentation. For PFAS, water concentration and grab sampling is similar to passive samples. Beautiful example. But how about the chemicals that aren't very stable? Oh. What about unstable compounds? From Anna Wang.
They won't be stable if you try to keep them in water. There is a possibility that chemicals that are not necessarily very, very stable in water, once they're absorbed into a solid phase extraction solvent or accumulated in a polymer, they might be more persistent, at least. But the problem of chemicals that degrade relatively fast, and that's an issue for passive sampling. But for a water sample, it's across the board.
Now for a tricky question, because it might involve fish biokinetics. It seems reasonable to expect chemical levels in fish to be greater than in passive samplers, and that the later may assess bioavailable chemicals, whereas the former may assess bioaccumulation and thus account for depuration. Do you agree?
So it's interesting because the graphs are presented. There isn't, for the moment, a clear explanation that's been validated. There have been some proposal for explaining the fact, because intuitively we would think, okay, the activity of bioaccumulative and biomagnifying chemical would be higher in the fish compared, especially at a trophic level of 3 or 4, compared with the water. But at the same time, or at least one of the assumptions is that when the chemicals start to accumulate at the base of the food chain, they don't necessarily, when a fish or something's eating algae, the concentration in algae has not necessarily reached equilibrium with the water. So things are being eaten through a food web at a rate that doesn't necessarily mean that everything is at equilibrium to start with. So the activity at the base of the food chain is a lot lower, and this means that we end up with concentration in fish. But we need to understand that. Yeah.
No, thanks for that answer. And I think I look at the time, and we're going to have to cut short. We might even get kicked off. We do have a few questions left, and we'll try to reach out to them. Just briefly, they did involve 2 passive sampling biosensors and HLB disk samplers. So we'll reach out to you for responses. And I'll just say thank you all for attending this webinar. Please note we have another webinar coming up on March 11th on the life cycle impacts of fluoropolymers. Go to zeropm.eu to find out about that and future webinars, and also our super final event to be held this Brussels at the Tone Hotel EU, September 8th and 10th. And for Sarah Hale and myself and our great presenters, Ian, Allan, and Emma Knight, I want to thank you for watching, and we hope to see you at the next webinar. Bye-bye.
Thanks, everyone. Bye-bye.
ZeroPM.
Zero pollution of persistent and mobile substances. This project has received funding from the European Union's Horizon 2020 Research and Innovation Programme under grant agreement number 101036756.